Welcome back. Please settle down. We are going to start with the Silver Jubilee Oration by Professor Gokhale. So would you all please come towards the front? Students, please come to the front. Don't be backbenchers, please. No backbenchers in science. Please come to the front. There are students in the balcony. Unless you want to stay there, please come down. There's enough space there. Come on.
देखिए गिरे ना भाई, बड़े लोगों का है।
Those who are standing behind are requested to kindly take your seats. So thank you very much for joining us after the lunch break. Now this is the time for the AIIMS Silver Jubilee Oration. I request Dr. Randeep Guleria and Dr. Chitra Sarkar to kindly chair this session. Dr. Randeep Guleria is former Director, AIIMS New Delhi, and presently he is the Chairman of Internal Medicine, Respiratory and Sleep Medicine and Director, Medical Education and Medanta, the Medicity. Welcome, sir. Professor Chitra Sarkar is currently President of AIIMS Kalyani and ICMR Emeritus Scientist. She formerly held the positions of Dean Research and Head of Pathology at AIIMS Delhi and President of AIIMS Guwahati. She is also a recipient of DBT National Women Bioscientist Award and JC Bose National Fellowship. Welcome, ma'am. I also request Dr. Rajeev Kumar, Associate Dean Academics, and Dr. Girija Prasad Rath, Registrar AIIMS, to kindly join the dignitaries on the stage. I request Dr. Girija Prasad Rath to kindly welcome Dr. Gokhale. Sir, sorry. I request Dr. Rajesh Gokhale, Secretary DBT and DST, to kindly join on the stage. Thank you, sir. Sir, Director, sir, please join us on the stage, sir. I request Dr. Rajeev Kumar to kindly introduce Dr. Gokhale. Thank you, sir.
Good afternoon and welcome to the 26th edition of the AIIMS Silver Jubilee Oration. This is now the 26th time that we're having an oration, and it's my pleasure to introduce the orator for today. The orator is Dr. Rajesh Gokhale, who is currently the Secretary for the Department of Biotechnology and holds an additional charge as Secretary of Department of Science and Technology for the Government of India. I think the best way of describing Dr. Gokhale's work and achievements is that he is a chemical biologist, and this is what his long-term colleague Anurag, my batchmate, told me. But that's the best way of describing him. He's got a master's from IIT Bombay, followed by a PhD from the Indian Institute of Science. And then he went on to do his postdoctoral work at Stanford University before returning to work in India. Most recently, he was director of the Institute of Genomics and Integrative Biology till 2016. He's known for his studies on the metabolic diversity of pathogens and is credited with the discovery of the family of long-chain fatty acid cyclic AMP ligases, and his studies assisted in the elucidation of the biochemical crosstalk between fatty acid synthesis and polypeptide synthesis in Mycobacterium tuberculosis. He holds US and Indian patents for his invention of methods to modulate pigmentation processes in the melanocytes. He's received a number of awards, and I'm just going to enumerate the most prominent ones among those. He was a Wellcome Trust International Fellow in 2001. He received the Shanti Swarup Bhatnagar Award in 2006. He was the Infosys Prize for Life Sciences in 2013, and of course, he's also a member of many Indian science academies. It's our pleasure and privilege to invite Dr. Gokhale to deliver his oration, and his oration will be titled Harmonizing Connections, Inter-Organ Crosstalk for Metabolic Health. Dr. Gokhale, please.
स्विच करना पड़ेगा।
Thank you very much for your very kind introduction. It's very happy to see very old friends here, Professor Guleria, Professor Chitra Sarkar, Professor Rath, Professor Rajeev Kumar, and of course, Director AIIMS and other friends whom I have known for the last 15, 20 years. And I think it's such a delight to be over here to speaking to you for this Silver Jubilee Oration. I think research and medical areas are so intimately connected that we obviously cannot think beyond getting good connections. And that's why I gave a talk of actually about harmonizing connections actually. And I think we need to harmonize connections not only to do good research, but also to get good value out of research as well actually. And I thought maybe I'll start by giving you a perspective of where the India is right now focused on. Does this work here on the screen? No. It works, okay. Thank you. So where does India wants to focus, and what is our target right now in this world? And I think if you really try and understand, it's a very interesting conundrum that is about middle income trap. You can see this chart is basically from a low income to a low to middle, low to income to high income, and becoming rich and staying rich. And I think the question that has come for several decades now, for several countries, you can see like Brazil, Mexico, South Africa, Russia, is why a developing country with a fast growing economy finds it difficult to eventually break into the ranks of rich nations. And this is a conundrum that in fact India will have to solve as well if we want to achieve the target of Viksit Bharat by 2047, what we're talking about. Now this is a challenge because unfortunately, many promising economies have become ensnared in middle income mediocrity actually. And you can see this is the middle income trap, and this is a concept was given in 2006 in the World Bank, and has been since. In fact, it's not proved or disproved, but it's just a phenomena that is actually observed, that very many countries find it very hard to move from this place to the becoming rich people actually. Now, of the 101 middle income economies, only 13 become high income by 2008 actually. So you can see it's not easy to escape these kind of traps and be able to do this. What we believe, and what government actually is completely on board, is saying technologically empowered India can only escape this trap. There is no other option actually Indians can work, particularly when we have to work for such large number of people. And the other very critical component that is actually still we are trying to struggling with is different trajectories are needed to leapfrog each quadrant actually. From reaching low income, so LMIC country, to the middle income is one trajectory that one needs to take. Unfortunately, I believe that this is, to go from here to here, you require a completely different trajectory. What that means is we really need to reset many things before we can actually cross over to that place actually. And how do we really achieve that target? And one of the aspects of this, I believe, for all of us to contribute is what I think is becoming incredibly important, is what I call intelligent networking actually. Very often we undermine our own performance primarily because we limit our networking to limited kind of people and areas whom we are looking at actually. This is an example that I really like the most actually. If you look at chemical principles, also define how interactions are actually very, very intelligent in its own self. If you look at hydrogen and oxygen, both of them are inflammable gases actually in some way or the other. But if you interact with each other, they in fact now are the ones that are used to actually extinguish the fire. So all these supporting materials in fact network, and therefore the effective outcome is very, very different actually. And that's what I think in the present era the real challenge is. How do we stay connected and stay relevant? And I think that's what is the very critical component, because the resultant communication determine your impact on surrounding society and the world. And therefore it is very, very important and critical that we actually learn to work together in a harmonious way such that one can create an effective trajectory that is very different and is an effective outcome of multiple vectors that actually give you a right directionality. I think Department of Biotechnology, just to give you a perspective, in fact, is trying to look at in this same space. And if one needs to grow the country, you really need to do this in many ways. You have to innovate, you have to generate infrastructure, you have to develop technologies, you have to support startups, you have to do financial models, inclusivity, social impact, sustainability. And all of these, in fact, every institution need to do. And every organizations and every individuals, in fact, also need to work in this kind of way. How do we really make this and drive this in the country such that bioeconomy becomes a relevant aspect of it? I'll just give you from the perspective of Department of Biotechnology. Over the years, it's a very small department, and all of you are well connected with it. Actually, in fact, AIIMS has a very good connections with it, but it's one of the smallest department in the government of India, but has done incredibly well, primarily because it has tried to put together different new steps that are ahead of its time actually. So for example, the first and foremost step that was taken, I thought was very, very ahead of its time, was in 2008, when an India alliance was created with DBT Wellcome Trust actually. The whole idea was, can we advance discovery and innovation to improve health? And the idea was, can we build capacity of the highest order in this country of people and researchers that eventually will make difference to the activities that we will perform in all the dimensions of areas. And in fact, it's good to see that 15 years, this has actually worked really, really well. If you meet any researcher all across the country who have been funded by India Alliance, they're probably very, very excited and happy about it. And I think we are going to renew this for another 5 years. In fact, looks like Wellcome Trust is happy to put in another 50 million pounds for next 5 years for this program. And I think in this program, we are going to be more collaborative programs rather than individual-based program, such that it can enhance the value for it. The second was the setting up of BIRAC, actually. Now, this is, again, an ecosystem building in its own ways. What this have done is set up industrial development and entrepreneurship as a major activity as part of biotechnology institutions, actually. And you can imagine, when this BIRAC was set up, in fact, country had about 75 startups in the biotech sector. And today there are close to 7,000 startups that are incubated all across the country. And you can imagine how institutionalizing processes, therefore, has a tremendous amount of impact in terms of building future goals of the country. In recent times, what we have now done is we have set up this organization called Biotechnology Research Innovation Council. Now, this is a network of DBT research institutions that will enhance and implement the transformative power of S&T for the benefit of all. In fact, we had 14 institutions all across the country, and all these 14 institutions, we've kind of put them together into one single sector such that they can actually work together in a cohesive way. And finally, which is going to happen very soon, hopefully this year, sometime in the middle of the year, we're going to set up what we call Mulankur bioenablers, actually. And this is to catalyze green growth and bioeconomy with PPP models, where artificial intelligence and biomanufacturing hubs will be set up all across the country that will in fact catalyze these activities to do. So you can see how planning and execution and strategic development is really critical for future of the country, and how can we create these new dimensions that will work. So a question, for example, always get asked, why do we need new institutions actually? And how should new institutions be set up if one wants to look at it? And you can see, why do we need BRIC? And one of the simplest way to look at it is that early on, when we were a low-income country, the whole idea and genesis of institutions all across the country was pursuit of knowledge, creating infrastructure, and capacity building. And these were the three paradigms in which all institutions in the country have been set up, actually. And therefore, they have done incredibly well, and we should pat our back for us to be able to be part of this ecosystem. However, in the rapidly evolving landscape of 21st century, every country thinks that science and technology and innovation is a critical driver of societal progress. And it's very hard to imagine how institutions can be directly driver of societal progress if we continue to work in this paradigms of systems, actually. So the institutions also now have to evolve into a new systems, as I said, to come out of middle income trap, to be able to work towards societal challenges and problems in a way that becomes a catalyst of change and development. And I think that's why it's very important to renew all the institutional capabilities, its capacities, and its ability to execute tasks in a way that it will make it impactful. So that's why I think the BRIC is going to be a transformative, and we call all the institutions of BRIC as iBRIC. And therefore, this is going to restructure and bring value. For example, the whole idea is to become the transformative power of S&T. So therefore, the very important aspect is to strengthen and align the scientific and technological innovation ecosystem, actually. There are a lot of fragmented efforts all across the country in various programs and projects. Can't we consolidate that? Can't we bring them together such that they actually become problem solving, then a project solving, actually? And somehow, this needs to be energized in the system such that everybody feels that I want to be part of the larger ecosystem, then rather than feeling that I am also doing something else, actually. And I think these institutions are largely going to be able to do that in its own way. The second is, India obviously wants to do global positioning now. We are no longer a country who wants to decide what others will tell us to do, actually. And therefore, bold global actions for driving bioeconomy does require critical strength. And I'm sure All India Institute of Medical Sciences would be one of the biggest player in this area as well. The third is asset monetization and management, actually. One needs to provide compelling incentives to nurture the startup ecosystem and be able to develop technologies such that they actually reach to the people. And that's the only way you can solve the problem. And the fourth challenge is obviously continuous capacity building. How can the workforce transitions will continuously evolve such that one can actually continue to survive in a space that is very beneficial. And for this, we are actually going to start a new program on BRIC RCV PhD program, which is we call I3C, which is Ideate, Immerse, Innovate, and Collaborate. And one of the activity that all the students who are enrolled in this program is going to do is in fact go out in the field for 3 months, and we have kind of provided, we will provide a fellowship to them to go out and work in the field, and therefore derive problems, derive ideas, develop questions, and come back and work in the lab on these questions that can make impactful final solutions for it actually. And I think that's why it's going to be a very, very enterprising PhD program, would be launched probably next, I think in this week itself actually. So the whole idea now that we believe is that novel innovations have to come together, and somehow rather than competition, we have to focus on cooperation and collaboration, and that's going to be the central piece of future of the country's progress. So obviously I've written 3 main agendas that DBT has on its own. Foster a future that is more sustainable, innovative, and responsible to global challenges. Potential to create new jobs and intensify entrepreneurial momentum, and Bharat to be among the global leaders in the next industrial revolution actually. If we work on these 3 large mega problems, we can in fact resolve many of the important challenges and crises to look at. It's good to see that biotech ecosystem is growing like amazingly well actually right now. In fact, last year, the growth of the bioeconomy has registered a growth of 29%. It's an unprecedented growth actually. The CAGR of 13 to 14% growth for last 3-4 years we have seen in this sector. So this sector is going to go very, very big. It's valued right now at 137 billion economy, and we believe that in 2022 it will be about, this is in 2030. 2047, we want to convert into a 3 trillion economy. And if we bring it to a 3 trillion economy, I think we'll probably be competing against the best of the world in countries to look at. If you really look at which are the sectors that are doing well actually, and you can see 43% corresponds to the bioindustrial sector, a very growing area, booming area, lots of enzymes, lots of new products, lots of new mechanisms by which you can bring in technologies to the place. Biopharma obviously is the second. Bioagri we would like to grow and make it bigger. The bio-IT itself can also grow very big, in fact with the AI and ML actually entering into the market. And the COVID economy still existed, 6% of it actually. So what are the challenges? How do we really work around these challenges to have what we call regenerative bioeconomy actually? And therefore, the key going to be in the future of the world is really to solve very, very big challenges and problems of forests which are burning, glaciers which are melting, and biodiversity's decline actually. And if one wants to look at the human history, the materials have been the pivotal role in the civilization processes. You can see every single material, whether it is metal, glass, cement, ceramics, fertilizers, or chemicals or plastic, every new material has transited into a new era of bioeconomy, actually, of various economical policies actually. And we believe the biomaterial is likely to be the most biggest transformative step that will work in the future of it. Therefore, at department, what we believe is that biology will build the next technology revolution in the future to come. For next 25 to 30 years, this will be the backbone of it. And therefore, we want to define for the department what we call BioVision for Viksit Bharat. And all of these sectors are likely to grow, and therefore we have sectorial mechanisms in which we are going to support these activities to be able to look at. For example, one of the major sectors that we are driving presently is what we call biomanufacturing actually. And the idea is simple, is that can we use the regenerative power of microbial systems or eukaryotic systems or the biological systems in a way that it gets economy to work on its own side. Maybe it's got to do with bio-based chemicals and enzymes. For example, our chemical industry produces as of now 70,000 molecules. And all of these molecules are what you call O2C market actually, so oil to chemical. Now can we convert some of them at least over next 5 years into B2C market actually, where we call biotechnology to chemicals, and this industry will grow like anything actually. The functional foods, as you can realize that agriculture is becoming a challenging everywhere with the soil quality going down, or the precision biotherapeutics, which is obviously going to be the future of the medical transformative care, climate resilient agriculture, carbon capture utilization, and futuristic marine and space research. And all these activities are the bio-enablers, what we call Mulankur enablers, that will actually grow in these systems actually. But what do we really need to do all this actually? And I'll just give an example through a simple poem that I actually really like to be able to put it out actually in front of you. And that is to do with the fact is only thing that we really need to do is to break the stereotypes what existed in our country actually. We all think that this is what we have to do, this is what we should do, this is what we can do. And therefore this is a nice example where it says, "Bhains ki vatha, bhains ki jaban se." And the poem is, बच्चा जब दूध थोड़ा बड़ा होता है तो दूध मेरा पीता है, वो भी बोनमीटा डालकर, और निबंध लिखने के लिए गाय, हाथी या कुत्ते क्यों? So you can imagine, you know, buffaloes and bhains are never treated well actually. You know, every time you write these essays, you write on these different aspects. अगर बच्चा लिख नहीं पाता है तो बोलते हैं काला अक्षर भैंस बराबर, तो क्या दूसरे जानवर पोस्ट ग्रेजुएट है? And you can imagine, you know, it's like one animal is always treated very differently, and I believe that's the problem in our. यदि कोई गलती करे तो लोग कहते हैं गई भैंस पानी में, तो बाकी के लोग क्या जानवर क्या कोको कोला में जाते हैं, और कोई ना सुने तो कहते हैं भैंस के आगे बीन बजाना, बाकी के आगे क्या लता मंगेशकर का गाना बजाते हैं, क्या बिगाड़ा है मैंने. So you can see it's a stereotype, right? It's a stereotype that we need to get out of to break this middle income trap actually. So let me transit here from department to more science a little bit, and I thought Vineet wanted me to speak a little bit of science as well actually. And so there have been transformative times of how science transits through various phases actually. And I think COVID-19 was transformative in its own ways, not just in a way what it caused morbidity and mortality, but what it reminded us of what really is critical component of scientific research is. And I believe one of the things that have come to the forefront is an inter-organ communication to the forefront of it actually. We've all become so specialized in our clinical practices, all our research practices, that we continue to focus on what we like to take a look at it actually. And we forget that all the other organs in fact are coordinatedly working through this. And in fact the most important and critical is organs talk to each other during normal physiology, and these mechanisms govern the homeostasis of organism. And therefore you can think of this as an opportunistic, or you can also think of, you know, real sense. Everybody's specialty talks about how COVID-19 and liver diseases, gut and COVID-19, chronic kidney disease and COVID-19, brain and COVID-19. So everybody is connecting with COVID-19 because it was an opportunity. But in fact, it's a real scenario. It's a scenario that says that every part of human body is in fact has a wonderful crosstalk. Can we understand it? And can we use this to the benefit of real systems that we are interested to look at? And in fact, my lab, before I got back to here, was interested in looking at this problem, which is called cachexia. It's a pathological syndrome, which is a pathophysiological syndrome, I should rather say actually, which is actually stated as a multifactorial syndrome characterized by an ongoing loss of skeletal muscle mass, with or without loss of fat mass, that cannot be fully reversed by conventional nutritional support, and is driven by variable mechanism of reduced food intake and abnormal metabolism. Now this is a very, very important aspect to look at from a quality of life that you want to bring in terms of systemic aspects of human life. In fact, every disease now has a characterized by, every chronic disease has a characteristic features of cachexia. It's either systemic catabolism syndrome with profound consequences of patient prognosis, and it is with all chronic illnesses, whether it's infection or malignancies actually. Now why is this happening, and how can we resolve these kind of challenges and problems? And in fact, the pathway that seems to be very, very important and working right now is the activation of JAK-STAT and NF-kappa B signaling, which is the downstream transcriptional regulation that can induce variety of catabolic pathways in muscles and adipose tissues actually. And that's been the signaling molecule that's been looked at it for a while to be able to understand. So the two aspects that our lab was interested to look at, one was cancer-associated cachexia, which is basically a kind of abnormality or malignancy that triggers all these activities, and the infectious-associated cachexia, because my past interest in tuberculosis that I have been spending for a lot of time, in fact. In fact, if you look at cancer chemoxia, there are cancer cachexia, there are really large number of research articles that are coming these days, actually, and it talks about malignant inflammation, tumorokines, metabolic mayhem, resulting in altered interorgan crosstalk and the systemic dyshomeostasis. Now, the whole challenge is, in the clinical world and the clinical research really, how do we reset all these transformed changes that are happening in the patient, actually? Because if we want to give quality of life, it's absolutely important that we just don't look at one aspect of it, but can we reset these altered inflammation-mediated cascades to be able to bring it back to people, and I think that's been our major interest of it. Obviously, the most celebrated of all diseases for this muscle wasting is the TB, actually. And in fact, it used to be called consumption with a cough before it started to call tuberculosis. It's rather interesting that TNF-alpha, which is a characteristic cytokine, in fact, used to be called cachectin, actually. And it was first named as a cachectin, and you can obviously see that fatigue and excessive weight loss and anorexia. So it's a rather complex issue of what it can do. In fact, in our country, for a very long period of time now, we have been supporting nutritional support to TB patients as a part of an activity which is Nikshay Poshan Yojana, actually. And I think this is implemented since 2018, and financial incentive of 500 rupees per month is given such that you can improve your nutritional status. I think it's great, but the question is, are these nutritional beneficial to people, actually? And I think, therefore, there was this recent ration studies also came out which suggested that an early weight gain in the first 2 months was associated with 60% of lower risk of TB mortality, actually, a work that was published in Lancet. It's rather interesting, but if you really look at the details of the data, you'll find that only 52% of the people end up gaining weight, actually, in the first 2 months of their part. And the question that you'll ask is that, why is that the scenario? Why, despite giving nutritional supplementation, you're not able to recover the part of it, actually? And in fact, this is why we believe that it's a vicious forward loop circle in TB pathogenesis, actually. You have an infection, you have deranged immunometabolic axis, systemic TB systems set up, then there is a dysregulated organokine network, you again go back to an immunocompromised state, and then infection continues to grow. And this is a cyclical models that have actually made these sustained survival of these activities. The question then to ask is that, how do you start addressing such complex challenges and issues, actually? And what in our lab at that time we had started, and right now I think my lab is almost closing down, but, but what we have started to do is, in fact, we thought that the liver is one of the major organ of immunometabolic hub to really start looking at from a point of view of how can we address the problem of liver because of its perspective, because it's involved in synthesis, secretion, degradation, and regulation of various organs that it is looking at. Also, it actually connects the immunological signal to the metabolic signal in the body, and therefore you can see large number of important activities are done by the liver. And can we address these problems in a way that we try and address the problem? Is the liver function important in TB pathogenesis? That's the question we started asking, actually. And the first question to ask is that, is there a literature on it? And of course, if you go back and look at some of the literature and talk to people in AIIMS gastroenterology department, they will tell you, yes, there are papers where you can find TB in the liver. The question is that, does hepatocytes get infected with TB? Or it is the Kupffer cells that are present in the liver that actually functions. So we did this experiment where we infected mice with the aerial infection, classical infection model, and then you harvest lung, spleen, and liver. And you can see classically lung infection in the blue, the golden color is the spleen, and the liver, in fact, always starts at the end of the 4th day, actually. And a 4th week. At the 4th week onwards, you will see liver infection in a constitutive basis in this aerosol infection model. Now, if you really ask yourself, this is a classical condition of a chronic condition of TB. Now, what is the patients that typically come to the clinic of TB patients are? And very often, the patients don't come in an acute stage, but in fact come in the chronic condition stage. The question to ask, does TB enters human liver very often, and are there actually consequences of it? So you can see this is the infected liver in mice, and you can obviously, all those who are very good at looking at H&E staining will say, oh, there's a lot of immune infiltration in the system. You can go back and do phalloidin staining, and you can see that in fact ectopic granuloma-like structures are formed in the liver at the end of the 8 weeks in a post-infection model in TB. Now this is aerosol infection model that all the people use in the lab to look at the TB research. And what we are only saying is that there are hepatocyte cells and there are non-hepatocyte cells, and Kupffer cells are the resident macrophages. And you would imagine that these macrophages may be one of the classical cells in which they will reside. So therefore, we went back and isolated hepatocytes, and you can see this is a primary hepatocyte stained with phalloidin staining, very large hexagonal architecture, classical cells. And you can then see that they are stained with, to show the validity of it. And you can go back and prove the point is that in fact, primary hepatocytes from the mice harbors antigen 85B, which is a marker of TB residing in them. So it's rather interesting to ask ourselves that how often does liver pathophysiological changes are in fact considered to be important in a TB pathogenesis, actually, if you really look at. And I think if you go back and see, and therefore we wanted to check the general most common albumin, SGPT, SGOT, and GGT levels. And you can see post 8 to 10 weeks, in fact, there are some increase in the mice as well. And in fact, there are some papers that talk about infection in humans as well, actually. So the question now is, and I'm not going to show this data, but does pulmonary TB patients harbor MTB in liver? And there are several autopsies that we collected from the hospital liver biopsies. Indeed shows that TB resides inside the human liver. So the question then is, how does MTB infection alters hepatocyte biology, actually? Because that's what we need to understand now. Now that we think that this is a critical component, and hepatocytes being a large number of hepatokines that are secreted by hepatocytes that coordinate various activities, how would that work? So we therefore do what is classical transcriptomic analysis, and you can see infected and non-infected ones are actually segregates in a PCA plot, indicating that, and the uninfected one, all of them are together at the same place. There's a PC1 variance of 27%, not very high, but it was large, good enough to be able to look at. And one of the key difference that you found was regulation of lipid metabolic processes. You can see that starkly changed. And several macroautophagy, vesicular trafficking, hepatokine secretions. And some of the hepatokine secretions that we have to still study in the lab, and I hope somebody else can study, in fact, we'll publish this, are lipocalin 13, GDF 15, FGF 21, and selenoprotein B, actually. And all of these, in fact, in an RNA-seq confirmed by data, is actually 2.4-fold higher. Now, if you look at the GDF 15, it's an inflammation-induced central mediator of tissue tolerance, and it's promote waste loss by enhancing energy expenditure in the muscles, actually. So you want to ask the question, are these kind of pathogenesis responsible for triggering changes in a patient that actually does not allow them to gain muscle mass, does not allow them to gain all these activities? And in fact, we believe that GDF 15 and FGF 1021 are very, very critical in terms of its pathogenesis in TB as well, actually. You can go back and identify them in the other aspect that we were able to show very nicely was that the lipid bodies are formed at a very large content, and this is driven by PPR gamma, which drives the biogenesis in hepatocytes, actually. The other very critical component was then how does MTB resident hepatocytes respond to anti-TB drugs? Because one of the questions that we will always ask that all the drugs pharmacology actually is affected by liver. And therefore, if you do look at this data, this is the rifamycin data, and this is the data for the isoniazid and the rifamycin. Oh, sorry, this is rifamycin, this is isoniazid. You can see for all the conditions, which are blue for the primary hepatocytes as well as HepG2, in fact, the drug clearance is faster, and therefore we believe that the resistance is being developed. Various drug metabolized enzymes, classically NAT2, which is the modifier of isoniazid, and the CYP2E1, which actually clears rifamycin. In fact, both of them are upregulated in the liver, clearly indicating that this aspect of liver metabolizing enzymes increase is what is altering the phenotype of the bug. And therefore, you can imagine that if you have a chronic case condition in TB, and if validated in humans, one could imagine that this is one of the common source of generating drug resistance bacteria, because you're providing suboptimal drug. Your drug is in fact being hydrolyzed very rapidly and transformed, and therefore you are going to have into a challenging situation where you're providing concentrations below the MIC, and you could develop these drug resistance pattern. In fact, this is the student who has done all the fantastic work, Vinayak Sarkar, along with Jyotsna. And I thought last 10 minutes, if I have 5-10 minutes more actually, if I can just talk about one program that we did it with AIIMS collaborations actually, and therefore I thought this is the right way to end the story. It's a very quick story actually. For all those who wants to go back and look at it, can look at this eLife paper, work done with Vineet Ahuja actually on looking at how liver regulates the intestinal colitis actually by modulating bile acid synthesis. And again, a very interesting program where again what we're trying to understand that how interorgan crosstalk is critical for relieving pathogenesis of various diseases. And as you all know, I don't need to give you details of it, but gut-liver axis is probably one of the very critical component of all diseases that are emerging. One is the bile tract, which liver produces bile and secretes into the intestine, and the portal vein that carries out the secondary bile and the dietary metabolites back to the liver. How does they work? So it's a very common model that is used in biologicals. All the labs, and large number of labs use this. It's called DSS-induced colitis model. Now, if you use DSS there, you can lead to mucin and goblet cell depletion, damage to junction proteins, infiltration of microbes, activation of inflex immune response, and enhanced intestinal bleeding. And damage to the colon is reversible in this acute model actually. So if you do this, you can then have, endotoxin going to the liver. And you can examine this by doing RNA-seq study of the liver, where you can pick up the lipopolysaccharide in the activated hepatocytes actually very easily, to be able to showcase that indeed, as you do DSS treatment, the liver is starting to get engaged into the systemic response that is mediated by the colon. Now, what does LPS does? And this is just a putative model that we set up. What LPS would do, activate TLR4, and then activate NF-kappa B and STAT3 phosphorylations, which will then are the inflammatory mediators and leads to secretory cascading event actually. So how do we look at this? So you can do the Western blot in the tissues, and you can see the phospho-relay actually getting activated on the day 2. Similarly, you can see phosphostat3, both 727 and 705 getting activated on day 2 to day 4 actually, clearly indicating that the liver is starting to respond to the inflammatory signals that are coming from the intestine gut. And this leaky gut now is going to engage liver in its pathophysiological consequences to be able to do this. To be able to look at this data, what we got was we in fact got these relay STAT3 knockout mice in hepatocytes actually. So you take these hepatocyte specific knockout mice, and then you do the same treatment and ask a question, what does happen? And we were rather surprised by this data, and with that time when we looked at it, what happens is that whereas the colon shrinking and all the disease activity index is there in STAT3 and the relay, but the double knockout mice in fact was recalcitrant for this infection, DMDS treatment actually, indicating that in fact the colon damage is less if you delete STAT3 and relay from the liver of the same mice. Now this is rather perplexing, because what you are talking about is loss of information is leading to gain of function phenotype, gain of disease phenotype actually. And therefore, to cut the whole story short, I will just go to the final results, since I'm already running over my time actually, is that the process that we found out which was differentially regulated was the bile acid pathway actually. And the bile acid pathway is not regulated by relay and STAT3. You would ask a question. How is it that relay and STAT3 is regulating bile acid pathway? Because it's an RXR pathway that is transcriptionally used to look at it. But in fact, I think we've gone ahead and done all the studies, and just to indicate that while the primary bile acids are always enhanced in the colitis model and secondary bile acids are low, the reason it has been always talked about is that the primary is not converted to secondary due to poor gut health, gut microbiota that is there. In fact, what we show in this study is what is happening, and this is the data from Vineet's lab, actually work done by a student, actually Manaswini. And I think you can see that both the cholic acid and chenodeoxycholic acid in the IBD patients is higher in the IBD cases in contrast to the normal patients. So higher cholic acid and chenodeoxycholic derived in the mucosal biopsies of IBD. Again, the whole hypothesis in this area is that the microbiome is getting defective because the secondary bile acids are not produced. What we are showing now, in fact, is that the liver, Relay and STAT3 starts to produce bile acids. The increased bile acid production, in fact, reaches gut, and it enhances the innate immune population, which is the resident tissue macrophages, lymphatic tissue dendritic cells, and also peripheral neutrophils, actually. There is no change in the helper T cells. And this then results in pro-inflammatory milieu, which enhances the inflammatory cascade, thereby leading to the enhanced indication of this. And you can show this by just using CDCA directly in the mutant mice. If you take a double mutant mice and inject CDCA, what we can show is we can replicate the same thing that happens in wild type, indicating that bile acids by itself, enhanced bile acid levels, can be the inflammatory aspect of biological process. Just to end the work, I think this is what homeostasis says. FXR signaling, cytochrome P450 is produced, STAT3 relay are inactive in hepatocytes because we show there are no differences in terms of mutant mice. You have an inflammation and there is no problem with the gut. You have inflammation, the relay and STAT3 are activated. The CYPs are produced in larger quantities. These two transcription factors, in fact, bind, and we have shown the data as well. And then accumulate PBA, damage the epithelium, and heighten the immune response, actually, and thereby resulting in enhanced damage. So you can see how long-range crosstalks are very, very critical for disease pathogenesis. And I gave you a couple of examples today, one to do with TB and one to do with other work. I think finally, I just have to thank large number of collaborators that I work with, Dr. Debashish Mohanty, Somen Basak Devram from NII, Dr. Vineet from here, Siddesh Kamath, who does mass spectrometry, Vinay Nandikuri has done all the transcriptomic work, and Nilesh Gaikwad, who have done the steroid mention. These are the students in my lab, and again, thank you very much for the opportunity, and I think congratulate all of you for this research program. Thank you.
Thank you, Dr. Gokhale, for a very interesting lecture. I now request the chairpersons to felicitate Dr. Gokhale, Dr. Randeep Guleria, sir, and Dr. Chitra Sarkar, madam. Thank you, sirs. Thank you, ma'am. I now request Dr. Tony Jacob to kindly take us to the next session. Thank you.
Thanks, Kamal. So Dr. Gokhale spoke about newer trajectories, and to lead us towards those newer trajectories, Professor Anurag Agrawal is with us, who will be leading a panel discussion on artificial intelligence, big data, innovative science, and transforming healthcare. Professor Agrawal is currently the Dean of BioSciences at the Ashoka University. He was formerly the director at IGIB. And he is going to first introduce the topic and then invite his panelists. So I request Professor Agrawal to take over, please.
Thank you all very much. I think I've become a high-frequency visitor to this podium, was here recently. Today I'll talk very briefly to set the context of a panel discussion on AI, big data, innovative science in transforming healthcare. Let's open the dialogue by introducing you to AIIMS. AIIMS is an articulate medical intelligence explorer by Google, and we have a presentation here. So one of the things as doctors we pride ourselves on, something that we would believe AI cannot do, is to have a conversation that is directed, like history taking, with a patient finally arriving at management, escalation, diagnosis, all of these components. So what Google did was build a LLM-based large language model, we'll talk about more about that later, that can do a text chat interaction with a patient actor. These are trained patient actors, and the problem is objectively structured case examination, OSCE. All of you are very aware of what OSCE is. More than 100 cases with doctors and patients from India, Canada, and UK were done. And they were assessed by expert specialist physician on the axis of management plan, whether the final diagnosis was in the top 3 differential diagnoses given by the program or by the human doctor, and by the patient actor in terms of perceptions of openness, honesty, and empathy. Now, basically, it's an AI competing with a well-trained primary care physician. At a very quick glance on this particular plot, and this plot basically takes every one of the axes and plots where you are. So you can see here, both types of people are open and honest. Both are doing good with top 3 diagnostic accuracy, management plan. What would you perceive is the area that AI needs to catch up? At a first glance, before I read the full legend of the paper, I was looking at empathy. But here's the reality. The pink is the AI, the blue is a human doctor. And these are full structured OSCEs. There is nothing except a text chat. It's not a clinical presentation where the doctor seeing the patient walk in will make the diagnosis by looking at their eyes. So that still remains, but for the vast majority of us, especially if you're thinking telemedicine, in this particular example, the AI was significantly better. And we had some clues on the empathy bit. In fact, if I think back to my own days, perhaps as an intern, never quite did a residency here, I can think back about empathy may not be the strongest feature when your clinic has 100 patients standing there. In this particular example, Rob Morris decided to use ChatGPT, this is version 3, and use them to counsel people looking for mental health support on a chat line. They were not informed that they were interacting with GPT-3, but as soon as AI came in, patient satisfaction actually went up. And the time to response went down. And one of the main things that was perceived, very empathetic, I guess because AI does not have a time pressure on its head. But the minute the patients got to know that they were talking to an AI, satisfaction dropped. You still want to talk to a human. When an AI tells you, I understand your problem, it doesn't actually make much sense. Let's take knowledge. And I'm again taking work from Google on a particular version called MedPalm 2. improvement over MedPalm 1. These are USMLE style questions. What I want you to see is how the accuracy has continuously improved and currently is around 87%. To those of you who have taken USMLE, you know how high 87% is. This is kind of like the topper of the exam. But more importantly, these are 1,000 questions. On the yellow is the AI, MedPalm 2 here. The blue is human, and both are being rated by an independent evaluator for good things and bad things. In every single one of the good things, the AI wins. The answer reflects consensus, reflects comprehension, better knowledge, better reasoning. Because they were asked to provide reasoning for the answers they gave. On this side are the bad things. More inaccurate, irrelevant information. The AI is worse. The AI does tend to produce more irrelevant information. But look at the more important one. Likelihood of harm. AI actually does better than the human doctors in this particular case. So we are at a very important turning point in medicine. And of course, it didn't come suddenly. This is a very famous cover, Garry Kasparov, 1996, 1997, Deep Blue. The brain's last stand. You know what happened? 96, Deep Blue 1, Kasparov wins. Deep Blue 2, 97, Kasparov loses. And Kasparov goes on to talk about how it was very unfair. It was all rigged against him. The computer was trained to play Kasparov, not play chess. But he ends his book, Deep Thinking, by saying he knew it was only a matter of a few more years. Go forward to 2016, 2017. And here you have the first AI system playing a game more complicated than chess, where expert players can't even describe why they do it. It's an art. They can't even describe every logic of a move, unlike a chess player. And DeepMind, which was later on acquired, well, by the time I think it was already acquired by Google, goes on to create a system that can play anything. Chess being one of them, but also Go, which was much harder. It beats the number 2 player in the world, Lee Sedol, quite comprehensively, 4-1. Now, go a little bit further to 2017, almost 20 years after the Deep Blue experiment. AlphaZero is born, given only the rules of chess, nothing else. Never gets to watch a game, can only play with itself. And it was easily the best chess player in the world. It could beat Stockfish, the program that can beat grandmasters. 64-36-0. For those of you who play chess, you know how astounding that is. And of course, staying with the theme of Google, because we'll have some experts later on, we also realized that this creates a new way of looking at things. AI is fantastic with images. I gave you the example of text and chat. That's the new area that's opening up now. But AI has been fantastic with images for a while. 2 critical papers, one in JAMA, diabetic retinopathy. I believe AIIMS also played a part in many of these evaluations. And more importantly, later on, Nature Bioengineering. How much information can you get out of a retina? I see Dr. Tityal is here, but I doubt Dr. Tityal, by looking at a retina, can tell me the gender of a patient. I'm very sure he knows the gender of a patient before he chooses or not chooses to look into their eyes, but I'm very sure he can't tell me the gender after looking at the retina. The fact is that AI can estimate age, gender, diabetes status, smoker, blood pressure, and CV risk. I've spoken to Nikhil multiple times about this. Perhaps the initial estimates may not be as good as originally thought, but the reality is it can give you all this kind of information from a simple retinal photograph. It will keep getting better. When I recently saw a Nature Medicine paper about how to use an ECG to predict cardiac risk long term, I was surprised, but these things are occurring. And of course, these things are very, very relevant in every possible setting, including poor ones. In Africa, when they had no doctors, they built a box in which patients could walk in, get an X-ray, and CAT4TB would give you a possible diagnosis of yes, no, and maybe, and some of those things could be taken away from human doctors. In India, qure.ai came up with a very, very good product. There are only 3 endorsed products by WHO. qure.ai QXR is one of them. And I've put one slide here because we again will have Prashant, who will be with us. And QXR by qure.ai can of course detect abnormal X-ray, can highlight abnormal regions, can identify common chest abnormalities and screen for tuberculosis. We will speak more during the panel discussion about this. A very important thing I want to point out is when it comes to images, there is almost no winning for humans, not in terms of finding things. If you were to look at an X-ray and develop an ontology tree, there are 127 clinical findings. If you were to now take thousands of X-rays and ask humans to find out the findings or the AI to describe the findings, in 117 out of 127, the AI is better. In the remaining 10, the AI is non-inferior. Now, please note, I'm not saying diagnosis. I'm saying findings. And this is a really important thing because there is context to everything. Now, an X-ray that looks like COVID-19 in one scenario could also look like heart failure in another scenario. To know the difference between the two and to have a prior probability estimate is what a doctor does better. But that now, with the first slide I showed you, in terms of the ability to have an interaction, figure out the differential diagnosis, then combined with analysis of images, is potentially going to open a brand new world. We are now at a point where a report can reach a patient in some parts of the world, this is Europe, without ever having been seen by a human doctor. In fact, many of the lab reports you get from lab equipment aren't actually really seen by a human doctor. You really think a pathologist signs a CVC by reading every part of it? Because the machines are very good. Here also, if a high quality AI engine says the X-ray is normal, basically saying there is no abnormal finding, perhaps there is no need for a human to look at it to find a finding. In fact, the human is more likely to be wrong if they found a finding on an X-ray, found normal by the machine. So at least in one country, Occipit was given the CE mark for the first autonomous AI medical imaging application. And this, of course, is on chest X-ray, featuring no abnormalities. If there were abnormalities, you send it to a doctor. Because as of now, nothing else is really ready for clinical practice. So with this, I'll come to the last part of what I'm saying. But despite my seeming to extol the virtues of AI so far in my talk, let's remember the most important fundamental of AI health. Health is the better half. It's not an equal marriage. It is not health for AI, it is AI for health. And the goal must remain to improve access, reliability, quality, affordability, equity, and justice. None of these are computer terms. While DeepMind got good at what it was doing by playing games, this is not a game. And massive amounts of data you need. So for example, if I remember correctly, the retinopathy work took around 128,000 labeled films before it got good. Of late, with GANs and synthetic data, we can use less and less. But clearly, we are using personal data. And at least in India, we are patient-rich, but traditionally, we have been data poor. We really want to get to a point where we are data-rich and hopefully disease-poor. And AI will be a very important part of it, but a better data ecosystem will also be a very, very important part of it. And I know Rajesh is doing a tremendous amount by IBDC, by many other initiatives taken by DBT. ICMR is setting up something at IISc to make data available. But we need AIIMS, many other top places containing possibly the largest amount of available data in the country to all come together to make a much better ecosystem. We are in the fourth wave, digital health. And to me, digital health does not mean conventional health delivered by digital means. It's a reimagination of what health means in the digital age. In this digital age, which is a convergence of both the digital and the genomic revolutions, what we think about health will change, how we deliver health will change, and we all need to get together to embrace this completely new reality. So thank you all very much. A parting thought, the meaning of it is relatively obvious, and I would now like to simply invite my colleagues, my panelists here onto the stage, and over from my side. I don't think we will do questions now. We will simply have all the panelists come onto the stage. I can introduce them one by one. And to start with, I would like to invite Rajroshan, because I have really been speaking about the work from Google so much. It really is most fair if I invite you up to the stage first. Professor Lipika De, my colleague from Ashoka University, and formerly at TCS doing natural language processing. Given all the stuff I praised about LLMs, we need some wisdom there. Krithika, who is very well known to all of you. Dr. Krithika Rangarajan I've known for years now. She's a radiologist trained here, but also has done, is doing, I always forget, I remind you, you're not submitted it yet, but her PhD in AI and machine learning at IIT-Delhi. Prashant, who has been the founder of the company I talked about, qure.ai. Again, the person who has probably truly brought AI to the masses over here. And Professor Anurag Rathore, whom I'm not able, there we are. Professor Rathore is right there. So Professor Rathore brings in a completely different flavor to what we have spoken about so far. He shows how big data AI can transform the thing that Professor Gokhale was talking about so much, biomanufacturing. The most important thing if India is going to escape the middle economy trap and truly become a rich nation. So with that, with those very brief introductions, each one of you is so distinguished that if I were to actually spend my time introducing you, the time would end here. So, Rajroshan, I will ask the questions in the same order that I brought in the topics. I want to ask you a question on data itself, particularly synthetic data. When I looked at AIIMS, it was a very, very clever use of searching the web, creating synthetic conversations. Pretty much, almost no personal data seemed to be used. It was all self-play. And I'm seeing more and more that compared to the retinopathy paper, where thousands and hundreds of thousands of patient images were used, recent papers use minimal amount of data. Where do you see this trend going? And are we going to come to a place where even without real human data in data poor states, we can actually do quite well?
That is an excellent comment. I think, am I audible?
It's not too well.
Is it better here?
Yes, much better.
Excellent. That's an incredible question, and it's something that has been a deep data science research problem, as you know. From the time that we published the paper on diabetic retinopathy in JAMA, it's about 2016, where, as you said, there were large amounts of data required for the model to perform. There's been a underlying research work stream that's been focused on how much data is good enough and how the model performs compared to the amount of data available. I won't go into the details, but one of the interesting findings was that beyond a point, the amount of data you have doesn't actually create meaningful improvements in the model itself. It's more access to high quality labels for the data. And then to your point, synthetic data is something that we're really, really looking at as an interesting area for augmentation at the moment. But as you said, I think a key part of the challenge with synthetic data, at the moment at least, is that it doesn't actually do a good job of simulating real world scenarios. So the model can sort of learn from a synthetic paradigm better, but if you compare it with the context and often the unstructured nature of real world data, I think that sort of element starts to go missing from the model's performance. So at the moment, I think the approach is to conjoin the two and see if the model can perform well. But to your point about, in the future, I think there is definitely a possibility where models will entirely be built on synthetic data, and then multiple models can link together. There could be a model that sort of looks at the output of the synthetic model and tries to understand if it is grounded, to your point earlier about real world simulated dialogue, and if it's actually understanding the sort of real world paradigm. So that model would be trained on more real world conversations, but the core model that's doing predictions might be synthetic. So it's a very interesting area of research, and I think that if you look at the trends of recent publications, you'll see that the data sets required are getting lower and lower. And therefore also the cost of producing these models, the cost of hosting them, the computational power is also lesser. So I think that's something that's really good for the field of research as well. So something that we're bullish about as an area of research.
One quick follow-up on that before I move on. So with the Privacy Act coming in India, GDPR in the Europe, and equivalent things coming in America, do you foresee that given access to medical records of institutions, for example, like AIIMS, followed by development of synthetic patients from those medical records, would that be a possible way to do privacy preservation while allowing AI developers to work?
Yeah, no, I think that's one area of research. There's a couple of different approaches we've taken towards privacy preserving machine learning and AI. I think one is, Dr. Tandon can probably speak to this better than I can. We've been working on this together for half a decade now, but you know, the fundus image in and of itself is, by most countries, not considered to be personally identifiable data. We did have an interesting conversation with the Singapore Eye Research Institute where they did consider it to be identifiable. But I think most of the initial data sets that have been used for research and development of the model on diabetic retinopathy, on the breast cancer models, the lung cancer models, they've all been de-identified patient data. So in that sense, no sensitive, personally identifiable information ever touches the model for learning and development. But in the future, to your point, you could imagine that de-identified data set merging with a synthetic data set to try and create more sort of high-performing models. But so far, I think our modeling has all relied on de-identified data, and it will continue to do so. I think that's key for us, not just with HIPAA compliance and GDPR, but also the developing data guidelines in India. I think it's one aspect is de-identified data, but the other aspect that's quite prevalent now is a likely geofence on the data not leaving India, for example, and all the research having to happen locally. And so that's another area of research to try and see how, if there is a locally trained model, can it then update a global model without sharing the underlying sort of data set. So that's federated learning is what I'm talking about. So there's interesting privacy preserving techniques that we're looking at for now. Very interesting question.
Fantastic. So we do know that we can preserve privacy while developing AI algorithms, and that's a great area to work. Professor Lipika, I'll come to you next. With your background in natural language processing and work on LLMs, you must be seeing now that LLMs and this entire area is become applied to things far different than language. In fact, pretty much it seems anything that follows a pattern can be tokenized. Where do you see these applications of LLM now going from visual transformers to text transformers going? And what do you see as the potential pitfalls we need to be careful about as we embark upon a new generation of generative AI?
So thank you very much for that question. We are already on the path to generation of multimodal LLMs. Again, MedPalm you referred to, we now are having MedPalm E, which is not only about language data, but also on visual data, sensor data, all of which are very primary data sources for medical decision making or clinical decision making. So the idea is that the representations that are created on the latent space is not coming only from the text, but they are coming with the associated images and sensor data, whatever. Now, multimodal research as such, multimodal predictive analytics, particularly with clinical text, is something which has been gaining field for quite some time, and it has been shown that using text along with the visual data or images, or particularly things like patient vital parameters, etc., have the capability to improve the accuracies of the prediction by quite a factor. So therefore, it is expected that these multimodal language models will be performing better in case of generating new elements. Now, coming to what are the new elements that we are looking at, it's something like if we go back to Dr. Gokhale's research, and he was searching for hypothesis and then testing it, imagine if generative models could be actually predicting many of those possible hypotheses or possible trajectories which could have been the cause of diseases, which are really the power of the generative models versus only the predictive models. The problems that we have associated with it currently is that how do we verify whatever has been predicted, just as probabilistic associations are also scientifically correct. And that is where a lot of work on how these LLMs can be stopped from, or how the hallucinations as they are known in terms of the predictive material can be really stopped or evaluated, and then some kind of confidence factors can be associated with the generations. So if you think about it, it's not very different from the chess or the Go playing activities that was always there, which is actually searching for a very large state space and then coming up with possibilities. The real challenge lies in how the additional fine-tuning and the training that goes on top of it to ensure that everything is also clinically valid and so on. So that's an area where a lot of focus is also coming along with multimodal LLMs, and that's agent-based solutions. So just like we don't have a single human expert attending to all the areas of decision making, the focus is, can we have decision support systems where each agent may be responsible for a particular task. So one agent is generating all kinds of hypotheses or possibilities. Another agent is validating whether that's really a correct hypothesis or not against scientific truths, maybe knowledge bases, maybe known grounded facts, maybe against cases which already exist to do. So essentially what we are really thinking of is a decision support system to begin with, where it's not maybe, as you mentioned, human beings are not yet ready to accept the decision given by an AI-based system, but at least it can be a decision aid for the experts who would have the entire chain of reasoning given along with a hypothesis to validate. And of course, then there is further learning to see how good these decision-making systems can be made. So that's really what I see as a path forward.
So would it be correct to say that in theory, with various ensemble of AI agents, in future we could even have a AI agent not just doing a text chat, but almost watching a patient come in and getting cues from multimodal information?
Yes.
Including the gait, the look, everything else like human doctors do.
Yes, that's also possible to verify and validate. Of course, that's one of the ways you get data, not only from what is stored, but also what is coming in right now. So, yeah, I mean, it's not just the pathology reports, the radiology images, but it's also a lot to do with the gait, the maybe the skin tone, etc., which can go in and into the decision-making process.
Thanks. Prashant, since I already praised AI and public health a lot in terms of deployment, I'm going to ask you the flip side. You have been part of massive deployments at the most peripheral of levels, including villages and districts in Rajasthan. Where do you see the challenges of deploying AI solutions in public health? And if there are challenges that are overcomeable in a sense of the word beyond data, where do you think we are going next with that? And do you see a point where in absence of doctors, you will have near autonomous actions by these public health deployable solutions?
No, that's a great question. I mean, great question. And I mean, just to also talk about our history, right? I mean, when we started Cure, we were building AI for interpreting radiology images. And we started with radiology because, just like you said, data is not easily available, and radiology data is always available. I mean, you have a lot of history. So that is why we started Cure in radiology and said, this is where we want to build AI. And initially the focus, I mean, when we started building Cure, the idea was to build solutions for the developed markets. And then we realized that there is a much bigger opportunity, there is a much bigger need in a lot of these remote places, right? If you go to remote parts of Africa, there is no radiologist available. Forget about Africa, in India also, right? You go to remote parts of India, there is no radiologist available. So that is where the need was. And our solutions were built for the developed markets. So we had to actually do a lot of modifications to make it work in the remote settings in Africa, for example, or remote settings in India and Southeast Asia. And many of these, right? For example, when we started building out, we said, okay, we'll send the data to the cloud, and the X-rays or the CT scans are going to the cloud, getting processed on the cloud, results come back. But the connectivity is very poor in many parts of the world, so that doesn't work. And then we had to actually install AI on a very, very small, I mean, sort of like put two mobile phones together, device of that size, right? We were basically able to install AI on a very small device and make it work in those regions. So that was one. So that was an innovation that we had to come up with, right? And I mean, and we had to build a lot of various other things. I mean, many times, I mean, there were, they wanted to actually, we wanted to digitize everything for our customers. So we built a mobile app which basically did end-to-end TB screening. So TB is one of the products that we have detecting TB on a chest X-ray, and end-to-end TB screening now is working on the Cure mobile app. So again, not only thinking about AI, but also thinking about how the AI integrates into the workflow of an end user. And I mean, Google does that really well, right? Google is probably the best AI company in the world, but you don't think about AI. I mean, everything is embedded. I mean, you look at Google or Gmail or Google Maps or whatever, right? Everything is super embedded into the workflow. So very similarly, in healthcare also, we have to think about how we embed that AI into the workflow. And especially in LMICs, especially in the remote areas, you have to really figure out what works there, because the workflow is very different. And you have a PAC system. If you, we work in the NHS, for example, there are PAC systems, there are RIS systems, EMR systems. There is nothing in many of the LMICs. So again, when you look at public health, the infrastructure is very different. So you have to figure out how you sort of integrate AI into that infrastructure. And that means that you might also have to build a bunch of that infrastructure as part of your solution. And it's not just the AI that you're building out.
Thank you very much. Would it be fair to say that as digital becomes more and more important in health, digital will become a determinant of health? Your access to digital, your digital public goods being built at a national scale might become a determinant of the health of a nation.
That's a very interesting question, actually. And it's true. I mean, I think digital will become a determinant of health. I mean, we are, getting more and more data about us online, on digital data, right? Including smartwatches, so many, everything. there are heart rate monitors, blood pressure monitors, glucose monitors, right? There's so many different things, or blood sugar monitors, so many different things that are collecting the data. And I think over time, a lot of that will be digitized, and you are now able to monitor people's health digitally and remotely. So I think definitely agree to what you said.
Thanks. Krithika, to you next. You would have tried to use AI in the setting of a tertiary care hospital with no real shortage of doctors and abundance of expertise. In these settings, where excellent people are already available, in your own area, for example, of reading mammograms or looking at breast cancer, or even thinking of solutions that go outside AIIMS, what has been your thinking and experience with how to best use these new multimodal AIs in tertiary settings where expertise is high, going downwards?
Right. So thank you, sir. So we've spoken a lot about how AI can really help in a low resource setting. But like you mentioned, even in a tertiary care setting, what AI can bring to the table is more than just detection to replace a doctor. For example, one of the things that we did was using a generative model where we tried to generate mammograms. These were completely synthetic mammograms. They're not mammograms of a patient, they're just generated mammograms. And we tried to say, can we actually get a control over what kind of mammograms we're generating? And what we found is that we could control the shape of the breast, the size of the breast, the density of the breast. And we said, what if now we can give a game-like tool to a resident to play with? And this resident had the option of placing a cancer anywhere in the breast. And how would that improve how the resident performs in a real world setting where he has to detect cancer? So this was one very interesting example of how we actually used AI in teaching the resident. And we found that the curve, the learning curve of the resident could dramatically be shortened by doing something like that. So we found that even over a 24-hour training period, which is nothing really, there was a significant increase in the number of cancers the resident was picking up. So rather than imagining things the way we traditionally do, like radiology teaching still is where you really tell somebody to imagine what a cancer would look like if it was in a different region of the breast. But here we were able to give an actual visual presentation of what it would look like. And that's just an example of what we can do in a tertiary care setting. But taking what we can do in a tertiary care setting beyond into the community is the other thing that we've been trying to do, like for breast cancer that you had just alluded to. So breast cancer happens to be the most common cause of mortality among women, even in India, although traditionally it's not believed to be so. It's believed to be a disease of the West. But despite having such a high mortality, there's almost nothing we offer to women to really detect the cancer early, whereas it is a cancer which has been proven to show an improvement in mortality if it is screened. So we've been exploring if we can just triage mammograms and say that this certain amount does not need to be seen by a radiologist at all, and then the rest goes to a radiologist. Can we actually increase capacity? And then we realized that even if we did that, we probably will not be able to screen the entire population, which is when we started building imageless ultrasound. So that's another work that we've been trying to do, where we can circumvent PNDT constraints to actually see if there can be a device which triages patients who can then be screened to make it possibly scalable.
Thank you. In fact, I would like to use that as an example of innovation, because only in India would this specific problem exist that you can't take an ultrasound out to do breast ultrasounds. So, despite AI, big data, despite sometimes shortage of data, an individual innovative idea will still find a place and traction. So that's a wonderful thing. So, Dr. Rathore, I would like to go to you now, a bit away from diagnosis and everything else. Amongst all of us here, you've used AI, very big data, even if not medical data, chemical data, to look at how to improve biomanufacturing, where the real growth of economy can come from. What has been your experience, and what should we be doing to better integrate all these things in India for the proper use of AI to stimulate biomanufacturing?
Okay, so thanks to Vineet and Anurag, I guess, for the invitation. So biomanufacturing, like Rajesh presented, if we are trying to catapult from this bioeconomy from where we are today to say 10 times higher in next 5 years or so, one thing to understand is that all of our manufacturing today is, none of that is cutting edge. If you look globally around the world, right? Almost all technologies that we use in biopharma or bioenergy for that matter are actually essentially copycats of what people do in US and Europe and so on, right? If we, and there are reasons for that, there are many reasons why it's like that. But if we want to grow our economy, to that extent, we need to modernize, right? And rather than just follow, and basically wait for innovations to happen elsewhere and then implement, we need to innovate. One of the concepts that, in the manufacturing world, which is relevant to Industry 4.0, I think I'm sure everyone has heard of, is the concept of continuous manufacturing, which is basically a fancy way to say assembly line manufacturing, right? So instead of doing things step by step and kind of wasting time in the middle, can I do it in a single go? Now, assembly line manufacturing is used for making mobile phones, for example, right? That's the reason, such an incredibly complex device is so cheap. and you can afford to buy it, for whatever, few $100s. In the biopharma, so far, very few Indian companies are investing in this thing. But if you want to go to continuous manufacturing, a big challenge is that you lose the luxury of stopping and thinking about, what's happening if something goes wrong, and then figuring out what to do to fix it. Right now we have that luxury because you're doing step by step. You see how this step works. You take your time. You decide, okay, now let me do this, right? In some ways, that's good because you have that time on your side. If everything is happening simultaneously, you lose that time. There's no time, basically. So what that dictates is you need to be able to do what we call as, real-time monitoring, which is basically if anything goes wrong, you have to know it right away. And then, you can't have a manual intervention to think about what to do. You already need a pre-programmed, solutions that automatically will tell the machine that, okay, if this happens, do that. And what is clear is that, AIML, just like I think you presented, right? Things like that, humans cannot beat machine in terms of speed, in terms of its ability to read, those tiny, tiny signals, right, that you would otherwise miss. And so the future really is that, AIML based, analysis of data. And by the way, these manufacturing plants generate huge amount of data. I mean, that's a huge amount of data set which is coming out of these plants. And so far, we are not really leveraging that much because companies look at it, retrospectively, they do an analysis and sort of like that. But with AIML, the future really is that the machines are looking at it in real time, and the machines are intervening again in real time. Though I must say that, I share all your enthusiasm, but I think there is an element of, at least so far, there is an element of human supervision, necessary. I'm sure all of you have seen the news from UK, where, our Rishi Sunak is having a huge problem dealing with, the post office there. The postmaster, the head of the post office got fired because apparently there was some software glitch because of which over a decade people got fired because there was a software glitch. So it was miscalculating, fund disbursement and stuff like that. Now all those people are being, reappropriated, compensated and all that. So at the end of the day, I think all these tools have certain caveats as well. Some level of human supervision, just to make sure things are going as they are, is always necessary.
Absolutely, and I would like to point out to the room that this idea of continuous manufacturing using all kinds of AI and other support was one of your key proposals in the Tata Innovation Prize. And these are ideas that are supposed to be able to transform a nation. So again, congratulations on that, Professor Rathore, and I really look forward to the idea of data being used for these manufacturing. You raised a very important point on these things creating trouble. I don't have a slide necessarily to show on this, but there was an algorithm that was used in America to predict who will become sicker and give them more care. And obviously insurance companies like these algorithms because who is your biggest friend? An insurance company that plans to pay. An insurance company that does not plan to pay, of course, is not your friend. But assuming they do plan to pay, they really want to keep you healthy. What they found is that black people would require a much higher value in terms of number of active disease conditions to get the same score compared to white people. Do we think the algorithm was biased against blacks or whites? Very, very unlikely. The most likely explanation is very simple. If the AI assumes that a sick person requires admission, all people admitted to hospitals or ICUs are equally sick, but one class of people, the poorer people, have to be sicker before they get admitted. The AI does not know that difference. So these kinds of algorithms turn out to be very racially biased, and that's a great example in the healthcare side, that there must be human supervision. There's a beautiful paper by Obermeyer. They were just happening to look at the data separating around racial lines, and they found massive systematic differences, which hopefully did not get anybody fired, but I think these are absolutely critical. I know that we should wind up a little bit earlier to get you all back on time, but I want to do one more round of questions on a topic that I believe will be very interesting to people in this room. The introduction of these kinds of topics, AI and data science, into biomedical sciences as part of the education. I'm not speaking only for medical, because there'll be enough people here who are not medical doing their PhDs. But I'll start in the same order that we went. Professor Day, what would be your thoughts about these kinds of exposures becoming an integral part of the biomedical curriculum? And have you seen efforts to do so?
Yeah, it's absolutely essential, as I have been myself working with, other biologists on their data, where I only understand data, whereas they provide the domain knowledge. And all the activities that were mentioned today can only go on with an understanding of the algorithms, what's happening at the back end, what are their weaknesses, and how to overcome them. And for that, it's absolutely essential that AI, machine learning becomes a part of the curriculum, even for students of biology. And towards what is happening, already I know in various IITs, machine learning is one of the core subjects that they have to do. So people from all disciplines, just as we have in smart manufacturing, also they have to understand AI and big data. Similarly for medicine, today a lot of chemical experiments are happening only through simulation. So everyone has to really understand it. So some core courses on AI, particularly machine learning, is already in. Maybe AI will also soon be one of the core courses to understand the decision-making process.
I'm looking really hard at my classmate, who's also the Associate Dean for Academics over here, Rajeev. So coming to you, Prashant, I'm sure you have been involved in deploying these things at the public health scale, which means you need to educate people about these kind of trends even further down the pyramid. How has that gone, and how do you think we should be sensitizing people towards these things all the way across the pyramid?
Yeah, I think it's a very interesting question. I mean, the reality is very stark. I mean, the reality is that people don't care. I mean, they get a result out. I mean, I'll give you an example, right? I mean, so you showed our chest X-ray interpretation, and it tells you everything. I mean, we have now 30 findings, 35 findings, right? So there is the X-ray. It tells you what findings are there. It marks out all the findings, and it produces that on the app on the phone, right? And we give that to every one of our users. We have maybe about 10,000 users across the world, and everyone gets that. Now, a lot of customers, in fact, maybe majority of our customers don't even want that, because their decision making is, does this X-ray have TB or not, right? That is one of the decision making. So, and just to also correct one thing you said, so for detecting TB on chest X-rays, WHO endorses to be fully autonomous. So we don't need a human reader anymore. So that means that once Cure says that the X-ray has TB, there is nobody else reading that X-ray. It goes to a microbiological test, right? So there are health workers now who are looking at the result, and they're looking at, they can look at the X-ray, they can look at the symptoms, all of the data is there. But the only thing they want is a TB yes or no. And we actually initially put that TB yes or no on the page where the X-ray is shown. And so they had to click, and they had to go into that screen and see the X-ray, see yes or no. But people want it on the first screen where the list of the patients is there, where it says, okay, this is TB yes or no, and they can decide to test that patient further based on that yes or no result. So the reality that we are seeing is that the more technology you provide, the more people become lazy. And unfortunately, that is an issue I feel with what we are doing in radiology, that the more we provide inputs to a end user, radiologist, or any other kind of physician, that skill set will go away. And I sort of link it back to Google Maps myself, right? That back in the day, we used to remember all the roads. We used to remember how to go from point A to point B. But then Google Maps came, it was super accurate. It can take us from any place to any other place, and you just follow the directions. And now we have lost the sense of direction. We don't know how to go from one place to another. And I've lived in Bombay for now 11 years, and I don't know a lot of places in Bombay. I would not have had that same experience if I was living in an era without Google Maps. So I think there is a very important factor that education is important, and definitely educating people on AI and computer science and coding is important, but also how do we make sure that people are not relying completely on AI? And this is a study that we are now starting to conduct with Gates Foundation, where we are trying to see how to empower the people to take decisions which go against the AI also, because they should not always rely on the AI, because AI could be wrong too. So this is a very interesting problem. I think something that we'll have to study over the next 5-6 years as we get more and more AI into not only healthcare but all kinds of decision making, right? And the issue that we have seen is that obviously if there is human supervision involved, then the AI is not very useful. The AI is useful if there is no human supervision involved. But if there is no human supervision, then how do you trust the AI to be fully accurate? So I think
a perfect catch-22. And with apologies to Rajroshan, who had nothing to do with the product, once in a while Google Maps can make you fall off a cliff as well. Professor Rathore, in chemical engineering, routine AI is anyway part of the curriculum. So to you, the question is somewhat different. The new age architectures, the idea of using embeddings and tokenizing anything from a genome sequence to a chemical reaction, how much do you think that needs to immediately come into training? Because many things like writing code are going to become much simpler, but thinking about how to tokenize is going to get much more complicated. So your thoughts?
Yeah. So, I mean, my view is probably a bit tainted because I am at IIT-Delhi, and so kind of a subclass of students is what I'm representing. I mean, our students, I think you don't have to tell them that AI is important. I think every student that enters IIT has already decided that they will do AI, ML courses, you know, irrespective of the branch that they've been in. That's not the problem. In fact, I see the reverse, at least for speaking for IIT again, it's a reverse thing that this craze for, you know, AI, ML as a way to enter data analytics, right? And simply because these are very high paying jobs, right? That's the attraction for an undergrad, by the way. You know, I think we are creating this generation of people who are, you know, maybe moderate to good data scientists, but I worry about the domain knowledge. And like I said, you cannot be successful if you don't have domain knowledge, right? And so at least I worry from an IT perspective that our graduates, you know, when they're graduating, say, from chemical engineering, do they understand chemical processes really well, right? Or if they are graduating from biotechnology, do they understand the biology really well? That's the challenge now, because frankly, they're not interested in these subjects anymore. All they are interested in is AI, ML, and data analytics.
Fantastic point. Reminder to the people in the room, you have to be a good doctor first. AI will come alongside. Rajroshan, coming back to, you know, interfacing between disciplines, something Google has done very well, cutting across with Stanford and many other places. Where do you see this intersection of medicine and AI growing, and what needs to be done for that?
Yeah, I think one of the first learnings we had when we started to apply AI in healthcare was it takes a village to make even the smallest impact. I think Krithika talked a little bit about using AI as a teaching tool, as a training tool. When we thought about designing, you know, technology that can have an impact, I mean, all of this was designed to be human-centered applications of AI in healthcare. I can give an example from Thailand. We started with the model for diabetic retinopathy, and you know, it was a really high-performing model on sense spec, so there was a lot of confidence that the model would generalize, which it did. From a purely academic standpoint, the model was on par with ophthalmologists. Started deploying that in the field and understood that the implementation challenges that come with deploying AI systems, the incentives for nurses and for practitioners, one of the most transformative learning processes for us was to understand how, you know, AI is one component of the system, but in many cases, you know, we've talked about bias in AI, we've talked about, you know, the human element there, the supervised learning, unsupervised learning, but just, you know, the humans that run the processes, what incentives and how they view AI is critical to how the overall system performs. In many ways, much, much more critical than the performance of the AI system itself, which for a company like Google, it's a very humbling exercise, right? Because at the end of the day, you believe that you come up with the best-in-class models and that will have transformative impact. It seems like a very binary outcome, right? But I think working with, you said Stanford, I would give examples of AIIMS here. We worked with, the governments in Thailand, interdisciplinary, but also I think multi-dimensional in understanding the design of a workflow that doesn't just involve the design of the AI solution. But how do you deal with, for example, we've deployed AI in remote clinics, in sort of vision centers, in community screening camps where there's no internet connectivity. So how do you then manage that sort of AI solution in an overreader capacity? How do you do batch uploads? How do you think about that process? And even in the Western world, where we think that there's a really, really sophisticated breast cancer screening system in the US, for example, it takes 2 to 3 months for workups to be done and for high-risk cases to be called back. So we did a study with Northwestern to try and understand if a real-time screening for sort of high-risk biopsy on the same day, for example, what kind of impact that could have. And again, it wasn't about the performance of the AI. It was if the system could handle that change in the standard of care and how the patient would perceive that from a health and well-being standpoint. So I think the learning has been the health economics, the health psychology, the workflow challenges. We remain committed to AI in healthcare, and I think that's, to your point, we've actually started investing so much more in the foundational models where you publish the embeddings. We were just having a conversation about this where we have CXR Foundation's work, we have Dermatology Foundation work, bioacoustics models where the embeddings are published, and then any research, any research team, any enterprise, any government can take that and build niche use cases that may not be commercially viable for companies to operate with, but the embeddings are good enough that the last 10-20% doesn't require a lot of expertise. So it's truly a multidimensional problem, and I would go so far as to say that AI is now, it's almost like a baseline. I don't think that in and of itself is any kind of differentiator. It's more about the domain learning, it's more about the operational challenges, and then the outcomes that you can measure from it, which ultimately ties to incentives in the system.
Thank you very much. And Krithika, I'll give you the liberty of choosing what you want to say to your colleagues in the hall. So no specific question to you. What are your parting thoughts for your colleagues here?
Thank you. I think that's quite a privilege. I just take a cue from what everyone was talking about. And I'd like to say this as a physician and a radiologist, that one of the first things you learn when you get into radiology is actually the physics of how that image is really acquired. So now that's not core medicine really, but it's really important to start there to really understand why an image looks the way it does and why it should represent a particular pathology. So with machine learning coming in, I think it's very much like the physics of machine learning that you need to know, because it's going to be here in the clinic when you need to sort of use it. And that would also help with looking at it as a magic bullet, which a lot of people do. So today, if you give people a report, they would think that it's gone through that magic bullet and you're supposed to do what it tells you to. But if you really understand the physics behind it, you probably know that it's just a probability and you need to work further and get the context to really get a final solution out of it. And also because there are a lot of medical students here and a lot of medical professionals here, just one thing that I'd like to say is that coding and machine learning is not quite as complex as it's sometimes made out to be, particularly in the era of tools like ChatGPT and the large language models, where it's automatically really helping you code. So I think a couple of the radiology technicians are here in the audience. There are some that who we've trained to actually code. They actually code. They write code with ChatGPT to really organize our data. So this is stuff that we can all do and is not something that we need to hold back from. And it should really be a part of, integrally a part of general medical teaching today, the way AI really is going.
Thank you so much, Krithika. With that, our side of this work comes to an end. It's a wonderful panel discussion. Depending on available time and where we are on the schedule, I can allow a few audience questions, but I look for guidance from either Vineet or in his absence. Okay, so let's have a few questions. We can have a show of hands and I'll try to call upon people. No questions, either we've been wonderfully clear or absolutely obtuse. Yes, please, Rajeev.
So you pointed a spotlight saying bringing AI into the curriculum and getting undergraduates taught AI. And there were obviously endorsements of it. The question is, how much do we teach the undergraduates? What do we teach and what do we not teach? Undergraduates, and we're talking of medical undergraduates, need to know computers, need to know coding, need to know AI, need to know anatomy, physiology, biochemistry, pathology, microbiology. Where do we stop? So the question always would be, what do we leave behind and what do we bring in? And if I go back to the question which Prashant probably brought up, saying I forgot reading numbers and phone numbers, which all of us did. I thought he's more my age than the young age that he looks. Domain knowledge. If you don't know what Krithika says is what is the physics behind an X-ray, why no AI, why no X-rays? Or maybe we'll reach a stage where we don't need to know them. Have we reached a stage saying all that you're taught is redundant, forget about it, learn how to run a machine, and you're done? Have we reached that stage? Or are we still, is it still necessary to teach them the basics? Is it still necessary to ask them what a lump looks like or feels like? Or is that now so redundant?
That's such a fantastic question. Of course, I have my own views, but I'm going to put Krithika on the spot because I have the privilege. So we're going to put Krithika on the spot of this one regarding how much is enough and whether no exposure at all to AI or how it works would be detrimental to a future graduate in time to come. So you can notice I flipped your question somewhat.
I think that's a very difficult question to answer, but I don't see what we can skip, definitely. I mean, I don't think there's anything we can skip. So clearly, I think we really have to keep training and, you know, our study days have to get longer. Yeah,
well, Krithika, I'm going to save you on that one. When biochemistry expands, we don't subtract something else. We simply add the new parts of biochemistry within the curriculum. So let me give my answer in that case. So my way of thinking of it would be that, you know, medical science has grown continuously while our number of years of training have remained exactly the same. We have managed in the past to squeeze in a huge amount of development within the medical curriculum without considering student overload. If fundamental exposure to what path AI would take, what are the areas where it is becoming near human in capability, where its weaknesses, would be instrumental in helping people make right choices. And number two, I cannot imagine a future in which physicians don't have AI assistants. What to trust, what not to trust, what are the true capabilities, could be covered in a relatively minor course which could be either done at AIIMS, could be done in collaboration with other people. It could be an elective, it may not necessarily even need to be compulsory. But for those people who can find the time, creating a structured exposure would probably be a good idea. I think I've already taken the 5-10 minutes I had. Maybe one more? Okay, one more question, please, sir.
Thank you. I wish it had continued for another half an hour or 1 hour. Yeah. So it was a very nice discussion. So I started some work in dermatology way back in 2018, and I got a lot of, you know, almost abusive messages from dermatologists, my own colleagues on the social media, and about their apprehension on job loss.
Yes.
So there is an apprehension that the specialties which are heavily dependent on pattern recognition, like dermatology, radiology, the many people would, you know, would become unemployed in near future or something like that. So is it something which is like transferring all blue collar job to AI and keeping cognitive work with us? That would help, or whatever steps would help alleviating this kind of anxiety or apprehension among doctors?
Thank you very much. And before I open it up, because this is a very important question, I will use a line I borrow from my wife, who is a radiologist, and who has a last slide saying, AI will not replace radiologist. Radiologists who use AI will replace the ones who don't. But I'm going to bypass Krithika on this one and perhaps go straight to Rajroshan. your thoughts perhaps on this, because I'm sure you also get this pushback from time to time as a company. So what do you think?
Yeah, we had a conversation about this, Dr. Somesh. I think it's fundamentally important, at least from the way that Google thinks about this, where we're looking at access to quality care. When we think about parts of the world, such as India, Southeast Asia, Africa, where there is such an acute shortage of expert-level care, I think there's a common tendency to start to look at a 1v1 comparison between AI and humans, especially, as you said, in pattern recognition tasks. But when we zoom out a little bit and we look at the space of healthcare, and we see all of the different nooks and crannies where AI is already adding value in workflow automation, EMR systems, data structuring, nobody seems to be threatened by those tasks, and they're all being done, as Prashant said, with Google Maps or Gmail or auto-suggest. Everyone's happy to search for cat on photos and look through all the fantastic patterns without worrying about what that's doing. But somehow, when it comes to performance on par with specialists, it starts to look at this as a binary outcome. I think it's important to think about the health ecosystem at large and see how many people don't have access to dermatologists or ophthalmologists or radiologists. And I was just talking to Apollo the other day, and we had a conversation about how 80% of radiology happens in these tier 2, tier 3 city imaging centers, where there is no access to quality care and there's no access to expertise. So in those cases, I think it's useful to think of AI as a funnel that can direct previously unmet needs or previous needs that have not been qualified to the system. And so we're not in any way, I think the AI is only as strong as the system it operates within. And if the system is bookended by experts and it's continuously used in an assistive way, I think there is room for both together to try and tackle more of the healthcare ecosystem than is being tackled today. And to your point, I think the radiologists with AI will probably outperform radiologists without AI. I think that is my thinking as well. And all of the papers we've done so far, I want to sort of mention this one very interesting cost-benefit utility analysis that the Singapore Institute did. They had three, arms of the research study, one with AI, one completely manual, and one semi-automated. And it turns out that the third arm was the most clinically useful arm for the particular workflow they were evaluating. So it wasn't just AI alone, it wasn't human alone, it was the combination of the two. And I think that will continue to be the case. I think even in the maps example, I would give it's something that I was going to bring up as well. I think the same way, but I also think that the fact that we're no longer worried about thinking about how to get from point A to point B means that while we're on that journey, we're freeing ourselves to think about potentially higher order problems to solve. And we have a lot of those to solve when we look at climate and health. And so I think there is an opportunity to use that lack of entropy with route mapping and convert that to some other sort of higher order thinking, if we choose to do so. I think there is an opportunity to just scroll on TikTok as well. But I think it's a choice that we as humanity have to make. And if we want to surrender to our base instinct and say, okay, fine, I'll just let automation take over and I'm going to just stop doing anything, that is one side of the story. And the other is, let's say, human will, which is always outperformed, will continue to think of higher order problems to solve. And there is so much more we have to do in that space. So I remain cautiously optimistic of the future.
And I would add one line that Professor Somesh Gupta built an AI tool for dermatological diagnosis, which was very much needed because Indian skin is very different from a white skin and things look very different. So such efforts are very much the need of the hour. So Professor Titial, last question to you.
I think we had a wonderful discussion. It seems future is totally AI-based medical healthcare. I had just one question, to the panelists and people sitting here. How would AI going to be its implication into a medical legal aspect? Would this save the doctors or physicians for future applications? Today we are under the cloud of our medical legal things. How would it help? It will be a positive thing or negative thing towards that?
Great question. And Prashant, since he creates products that are used by Indian doctors, can perhaps shed some light onto what the situation is.
I think regulations are a long way behind, and we are deployed in many countries where we are providing our assistance to radiologists and other physicians. But the reality is that regulations which will make AI fully autonomous, even you showed the occipit example, we have the same clearance, we have the same thing, right? But that does not allow us to sign off on a report yet, right? And I think one is the ability of the AI to be able to sign off on a radiology report completely automatically and be that accurate. And the issue there is that the burden of accuracy on AI is probably 100x more than a human, right? So that is true for autonomous cars also, by the way. If you look at autonomous cars, they are way more safe than human drivers. But there was, San Francisco launched autonomous cars, and within a month or 2 months, I mean, one of them got shut down, right? And that was because there was an accident and one person died, and they shut down that particular company from driving autonomous vehicles on San Francisco roads. So very similarly, I think, I mean, and that is true also. I mean, AI can have very systematic biases, and it could be wrong on the same kind of condition, same kind of disease many times. So you have to be very careful about making AI fully autonomous. So right now, I would say regulations around AI being autonomous, not only in healthcare, but in other industries also are not there. I mean, I think it's very restricted. But I think finally, I mean, if AI becomes autonomous in certain parts of the world, it is useful. For example, detecting TB on a chest X-ray, where we are autonomous, that is a need where there is nobody else who can fulfill that need. So then it's a question of which evil do you choose? I mean, not being able to read a scan or get AI to read a scan, which may not be 100% accurate, but it's close to getting accurate. But coming back to your question, I think regulations are still fairly far away from being autonomous and AI being fully able to sign off on a diagnosis or on a report.
A smaller version of what Prashant said, for the time being, AI will say, for clinical correlation and decisions. Thank you all. Thanks to the great panelists.
Please stay on stage for a while. That was a great panel discussion. Thanks a lot. And kudos to the research section for having organized this panel discussion, which addressed the entire spectrum of the audience, the misapprehensions of the seniors, to stimulate the juniors, as well as for those of us who are tentative to at least know something about it and know more about it. So that was a great panel discussion. We would like to felicitate the panelists with a token of our appreciation, some pretty plants that you can take back and put on your office sill or at home or wherever you want to take it. I also request Professor Titiyal to come on stage to felicitate the panelists. It doesn't stop with the plants. There are more goodies coming. So please. So there are more things. So thank you very much. So while you came into the auditorium, you would have seen that there are some 600 odd posters that are displayed outside. And these were judged last evening. And over to the award ceremony for awarding the best posters. And for that, I hand over to Dr. Sunita and Dr. Soumitra. Please.
Thank you, Dr. Tony. Now I heartily welcome you all to the final event of the day, the award distribution ceremony. It goes without saying that the award distribution ceremony is amongst the most treasured occasions on Research Day. It is an opportunity to celebrate the success achieved through sheer hard work and perseverance on both the part of the student as well as their mentors. To encourage a competitive spirit, the research section this year is giving about 88 awards in various categories, plus there are about 60 appreciation awards. I request all the dignitaries present here to please present the awards to the winners today. May I request, I think Director Sir is not here. May I request Dr. Subrata Sinha, Dean Academic Dr. K.K. Verma, Dean Research Dr. Tityal, Associate Dean Dr. Rajeev Kumar, and our guest Dr. Anurag Agrawal to please bestow these awards to the winners.
A very good evening, everyone. It's been a long day, but this is the time to get the rewards. So the way we're going to do this is we're going to announce all the prizes category-wise. So there are different categories like the undergraduates, the masters, MSc, PhD, junior residents, senior residents, nursing, project staff, SDG posters, innovative devices, and the selfie awards. So we first begin with the undergraduate category. And the first awards are the oral paper awards. The third award goes to Rutul Chag from the Department of Physiology. The second award goes to Akansha Singh from the Department of Physiology. The first award goes to Ishan Gupta from Gastroenterology. We now move on to the poster category. The third poster prize goes to Kamal Kant from Dr. RPC. He's a BSc student. Undergraduate. Kamal Kant. The second prize goes to Sushmita Somagattu from the Department of Obstetrics and Gynecology. And the first prize goes to Hayagribas MB from the Department of Neuropathology. There are two commendation awards. Ishan Gupta from the Department of Pathology and Madhur Singhal from the Department of Pediatrics. Next, we move on to the Popular Choice Poster Awards. The third prize again goes to Kamal Kant. The second prize goes to Ishan Gupta. And the first prize goes to Vaidehi Jha.
The next category is the MSc Awards. For the poster category, we have the third prize to Mahak Farhan, Biochemistry. The second prize goes to Arka Diti Gorai from Biophysics. And the first prize for the poster category goes to Munib Parvez from Biochemistry. We also have 2 commendation awards in this category. Anish Kumar Chauhan from Biochemistry and Sana Ainam from Biotechnology. In the MSc category, we also have 3 Popular Choice Poster Awards. The People Choice Poster Awards, the third one goes to Prachi Chaudhary from Physiology. The second prize goes to Unnati Sharma from Reproductive Biology.
Audience, kindly applaud the winners. They've really put in a lot of hard work and they deserve a big round of applause.
And the first Popular Choice Poster goes to Kajol from the Department of Anatomy.
Moving on to the PhD category. In the oral presentation category, the third prize goes to Manvi Arora from the Department of Biochemistry. The second prize goes to Minakshi Mendiratta from Stem Cell Facility. And the first prize goes to Jaya Tuck from Pulmonary Medicine. In the poster category, the third prize goes to Shruti Mahapatra from Stem Cell Facility. The second prize goes to Swarandeep Singh from Biochemistry. And the first prize goes to Mohini Mendiratta from Medical Oncology. There are 3 commendation prizes. Neelam from Biophysics, Alisha Arora, and Alamkrita Singh from Biophysics. Alankrita Singh, it's not around, okay. Moving on to the Popular Choice Poster Awards. The 3rd prize goes to Purva Bhargava from Pharmacology. The 2nd prize goes to Shweta Sharma from Physiology. And the first prize goes to Aarti Srivastava from Pharmacology.
Move on to the junior resident category. For the oral presentations, the 3rd prize goes to Dr. Hana Karim from Pediatrics. Second prize goes to Dr. Lanu Mona from the Department of Biochemistry.
Do we know who's
And the
getting an
first prize goes to Dr. Sona Fazil from the Department of Biochemistry as well. For the poster, in the poster category, we have a tie for the best poster 3rd prize between Dr. Philji George and Dr. Mistry Deepayan. Dr. Philji George, Department of Anatomy, Dr. Mistry Deepayan, Anesthesiology. The second best poster goes to Dr. Madhavi Nayyar from the Department of Physiology. The first prize goes to Dr. Kavya V. Nair from the Department of Anesthesiology. Dr. Kavya Nair. We have the commendation awards for the JR category. Dr. Sagar Rathi from Biophysics. Dr. Ripamchisa Sangma from Microbiology. Commendation Award for the Junior Resident category. Dr. Majage Siddha Vivek, Anesthesiology, Pain Medicine and Critical Care Medicine. Dr. Arka Dipta Mukherjee from the Department of Pathology. Awards for the JR Popular Choice category. The third prize goes to Dr. Ravi Kumar from the Department of Medicine. The second prize goes to Dr. Radha Rani Seelam. Dr. Vanshika Arora from Dermatology gets a first prize for the Popular Choice poster.
Now moving on to the senior resident category for the oral presentations. Oral presentation in the senior residence category. The third prize goes to Dr. Monica Endrakanti from the Department of Pediatrics. The second prize goes to Dr. Shubhankar Datta from Radiodiagnosis and Interventional Radiology. And the first prize goes to Dr. Akhil Santosh from Medical Oncology. Moving on to the poster category. The third prize goes to Dr. H. Anku from Dr. RP Center for Ophthalmic Sciences. Please give him a big round of applause. Poster category for the senior residents. Third prize, Dr. H. Anku. There is another third prize, Dr. Sainath Viranki. Second prize, Dr. Sherin Jacob. First prize, Dr. Shareen Verma from Microbiology. We have 5 commendation prizes as well. Dr. Vimmi Gautam from ENT. Dr. Anjali Krishna K from Anesthesiology. Dr. Archana Sasi from Medical Oncology. Dr. Ria Shriyan from Genetics. Dr. Sagnik Biswas from Gastroenterology and Human Nutrition Unit. Dr. Sagnik Biswas. Moving on to the Popular Choice Awards, the third prize goes to Dr. Priyanka Das from Obstetrics and Gynecology. Second prize, Dr. Nidhi Gupta from Biochemistry. And the first prize, Dr. Ankesh Gupta from Medicine.
For the nursing category, in the posters we have the third prize, which goes to Payal Kahol from the College of Nursing. The second prize goes to Vishakha for the poster category, Vishakha College of Nursing. The first prize goes to Komal Antil from the College of Nursing. Akriti Chaudhary for the Commendation Award. The second Commendation Award for the College of Nursing goes to Ms. Sitara Ilyas. For the College of Nursing, the Popular Choice Poster Awards are being given to the third prize to Ruby. Second prize goes to Shubham Gaur. First prize goes to Neelima Raj.
We now move on to the next category of project scientist or project staff. In the poster category, the third prize goes to Abhinav Saini from Biophysics. Abhinav Saini from Biophysics. Second prize, Anjali Gaur from Biochemistry. First prize, Dr. Bibhu Datta Mishra from Gastroenterology. We have 3 commendation prizes. Dr. Yaklesh Gupta Neeti Nadda, and Chandani Suvarna. नहीं, उसके बाद SDG है। We now move on to the Popular Choice Awards in the Project Scientist Staff category. The third prize goes to SK Datta from Anesthesiology. The second prize goes to Komal Kashyap from Onco Anesthesia. And the first prize goes to Reena Chittora from Physiology.
In the special category of the Sustainable Development Goals based theme posters, we have the third prize going, there's a fourth prize as well, which is the Commendation Award, which goes to Kanchan Sani. Kanchan Sani, Department of Biotechnology. The third prize goes to Lipika Silaj, Department of Anatomy. The second prize goes to Mamta Puruswani from JPNATC. The first prize for the SDG category goes to Dr. Rasna Praveen from JPNATC. For the popular choice poster in the SDG category, we have Kriti Negi from Biotechnology. The third prize goes to Kriti Negi, Biotechnology. The second prize goes to Dr. Mamta Puruswani from JPNATC. The first prize for the popular choice poster goes to Dr. KP Kochar, Physiology. In the Innovative Devices category, the third prize goes to Dr. Gopi Shankar. Dr. N. Gopi Shankar. The second prize goes to Dr. Subhash Chandra Yadav. The first prize goes to Dr. Sushma Sagar. First prize in the innovative devices goes to Dr. Sushma Sagar.
We now move on to the much awaited selfie award. Let me give you the numbers that we received. So the total research posters that were put up on display was 577. The total votes for the posters that we received is a staggering 10,000 votes. The total number of voters, 5,720. That's the total number of votes. And the total selfies posted, 2,511. I think we all deserve a big round of applause. Now announcing the winners. The third prize goes to Moses Azaria with 130 votes. The second prize goes to Ayush with 199 votes. And the first prize goes to Sweetie Rani with 295 votes. A special thank you
Rakesh Garg and Dr. Surinder Sharawat and their team for thinking of this brilliant idea, which encouraged a lot of audience participation. Thank you so much.
So we're also giving away appreciation certificates. That's roughly 1 in 10 posters. So you'll be, you can collect the certificates from the research section later on, but I'm going to read out the names. Under the undergraduate category, in the undergraduate category, Parnika Nangla from Pathology, Archisman Mazumdar, Hardik Gupta. In the MSc, Shikha Roy and Nikita. In the PhD category, Nisha Manav, Vishal Basu, Umar Muhammad Sheikh, Anurag Singh, Manisha Supriya, Diksha Patel, Iman Dandapat, Amber Rathore, Vikas Kumar, Ravi Chauhan, Bhaskar Sharma, Deepak Vats, Sanskriti Rai, Vivek Nair, Deepa Singh, Amit Khatri, Diksha Madhuri, Kanchan Saini. In the JR category, Dr. Anam Anil Alwani, Dr. Rekha, Dr. Samyadeep Datta, Dr. Madhan Vishal R, Dr. Navodit Sai, Dr. Nil Yadav, Dr. Akhila Gopakumar, Dr. Mudit Agrawal. In the senior residence category, Dr. Apurva Kabra, Dr. Arshi Tandon, Dr. Kiran Kumari, Dr. Muhammad Tabish, Dr. Shubham Sawhney, Dr. Chitrakashi Nagpal, Dr. Kanupriya Bhatia, Dr. Sakshi Ojha, Dr. Ankesh Gupta, Dr. Sheetal Agarwal, Dr. Amitesh Khare, Dr. Madhav Kirti. In the project scientist staff category, Shraddha Kapoor, Dr. Deepika Lal, Rachna Verma, Sonali Sen Gupta, Dr. Muhammad Tariq, Swati Pundir, Arpana Srivastav, Muskan Chadha, Dr. Pallavi Tiwari, Sushila Dahiya, Ishita Dhyani, and in the SDG, Kriti Negi, and in nursing, Shubham Gaur, Priya Singh, and Parul. Thank you.
Many congratulations to all the winners, and all of you who have participated will also receive their e-certificates through email. Thank you, sir. Thank you, ma'am, for giving away these awards. And we will have a formal vote of thanks, but I would also request you all to please stay back for a photo session. Thank you. Now we hand over to Dr. Kamal and Dr. Lata for a formal vote of thanks.
Thank you, Dr. Sunita. And Dr. Sumitra. So, ladies and gentlemen, good evening. We stand before you today with a deep sense of gratitude as we come to the close of this remarkable third annual AIIMS Research Day, celebrated on 29th and 30th January at AIIMS New Delhi. It has been 2 days filled with insightful presentations, engaging discussions, and the celebration of excellence in research and innovation. Firstly, we extend our heartfelt thanks to Dr. M. Srinivas, Director AIIMS, for his visionary leadership and unwavering commitment to fostering excellence in research and academia at this institute. A heartfelt thanks to Dr. Kaushal Verma, Dean of Academics, Dr. J.S. Titial, Dean of Research, Dr. V.L. Kumar, Dean of Examinations, Additional Director Administration, and Senior FA for their inspiring leadership and the tireless efforts in promoting research excellence at AIIMS. We express our sincere appreciation for the invaluable guidance and support provided by Professor Rajeev Kumar, Associate Dean Academics, Professor Naval Vikram, Associate Dean Examination, and Professor G.P. Rath, Registrar.
This year we have received an impressive count of 600 abstracts and several innovations. A big round of applause for our students and staff researchers at AIIMS whose participation made this event an interesting and successful event. We express sincere thanks to the abstract committees across all the categories, such as undergraduate, PhDs, junior residents, senior residents, MSc students, scientists, and project staff, and nursing staff who has spared their time from the extremely busy schedule to evaluate and judge these 600 submitted abstracts. I mean, completing this task on time was a tough task for everyone. A big round of applause for them also. We are very grateful to the committees that evaluated the AIIMS Excellence Research Awards and Critics Awards too.
A warm thank you to our distinguished session chairs today, including Dr. Y.K. Gupta, Dr. V.L. Kumar, Dr. Neena Khanna, Dr. Shinji Bhatnagar, Dr. N.K. Mehra, Dr. Subrata Sinha, Dr. Randeep Guleria, Dr. Chitra Sarkar, Dr. K.K. Deepak, Dr. Jayanta Bhattacharya, Dr. Rama Chaudhary, Dr. Meenu Vajpayee, Dr. Kaushal Verma, and Dr. J.S. Titial, sir. Your guidance and expertise ensured the smooth conduct of this session, making the academic exchange truly meaningful. We also extend our gratitude to Dr. Rajeev Behel, Secretary to the Government of India and DG ICMR, for delivering the AIIMS Golden Jubilee Oration. A very special thanks to Dr. Rajesh Gokhale, Secretary of DBT and DST, for delivering the AIIMS Silver Jubilee Oration. We are honored by their presence and the valuable insights shared with us.
Transitioning to the extraordinary team that orchestrated this grand spectacle, we want to express our deepest gratitude to the chairpersons, co-chairs, and scientist coordinators of various organizing committees, Central Committee, Poster Exhibition Committee, E-Directory Committee, and a special mention to Stage Management Committee, Media Committee, Catering Committee, General Arrangements Committee, and Innovative Devices Committee for their kind support and guidance for smooth conduct of the scientific session as well as the conduct of the research day. A big thank you to Mr. Yogesh and CMET team, Dr. Pooja Gupta and Computer Facility team, Engineering Services Division, Security Department, Sanitation team, Cafeteria, and Auditorium team. Your collective dedication and collaboration were the key ingredients in this successful event.
A special mention must be made for Mr. Uday from research section who worked tirelessly day and night, always sporting a contagious smile. Finally, I would request a standing ovation and heartfelt gratitude to Professor Vineet Ahuja, the maestro behind the show, whose meticulous planning, leadership, and a cool-headed approach made this event an unparalleled success. Professor Ahuja's ability to navigate hurdles, keep the team motivated, and maintain composure amidst a ringing mobile phone and a demanding clinical commitments exemplifies his extraordinary dedication and leadership. We extend our deepest appreciation for his invaluable contributions, without which this achievement would not have been possible. Thank you, Vineet sir, for your unwavering commitment and outstanding leadership.
We express our gratitude to the teams involved here, like research section team, team CCRF, team CRU, and team CMIE. Thank you, everyone. Lastly, as we conclude this event, we want to express gratitude to each and every participant, attendee, volunteer, and member of the organizing committee. Your collective efforts have made this event a resounding success. Thank you once again, and let's carry forward the knowledge gained and connections made to further enrich our endeavors. We look forward to meeting again next year with a more vibrant and exciting AIIMS Research Day.
So I now request you all to kindly remain stand up for the national anthem. Thank you very much. Please stand up for the national anthem.
Jana Mana Adhinayak Jaya He Bharat Bhagya Vidhata Punjab Sindh Gujarat Maratha Dravida Utkala Vanga Vindhya Himachala Yamuna Ganga Uchala Jaladhi Taranga Tava Shubha Name Jage Tava Shubha Ashish Mange Gahe Tava Jaya Gatha Jana Gana Mangala Dayak Jaya He Bharat Bhagya Vidhata Jaya He Jaya He Jaya He Jaya 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 He.
Thank you very much.
Thank you, everyone. Thank you so much.